hopefully that works. All right. So neurophys and myophys. Might as well just get started. All right. Can everyone see that? Yeah, I assume everyone can. Cool. All right. So basically, we want to know a little bit about the divisions of the nervous system. You guys know this stuff already. Split into central nervous system, which is the brain and the spinal cord, and also the peripheral nervous system. So somatic nervous system is all your conscious stuff, so your conscious sensation as well as your conscious movement. Then the autonomic nervous system is all the automatic stuff, and that's further split into sympathetic and parasympathetic nervous system, where the sympathetics are the kind of the fight and flight, parasympathetics are the slow down, rest and digest. And a good way of remembering that is that parasympathetic parachute, parachutes slow you down. So parasympathetic would be rest and digest. Okay, so the CNS, it takes afferent signals. So it receives afferent signals and it processes them and then sends out efferent signals. And a good way of remembering that is like the difference between affect and effect. Affect is like a verb you do to something. So you receive those signals. And then effect is what you do. So like the effect of that is that you do something to it. Hopefully that makes sense. I probably just confused a lot of people. So basically, we also need to know a little bit about the different parts of the brain. So there's this cerebrum here, which makes up the bulk of the brain. Then there's the cerebellum. Cool. And then there's the brainstem as well, which controls all the automatic movement or automatic processes. And then a little bit more in depth, you've got this is like the different folds in the cerebrum. So you've got a gyrus, which is kind of the exvagination, and then the sulcus is like a trough. So these are the different parts of the brain. So you've got cerebrum, cerebellum, brainstem, frontal lobe, parietal lobe, temporal lobe, and occipital lobe. Frontal lobe is pretty easy to remember. It's at the front. The rest you just have to know, especially for next year. These are the different functions of the different lobes in the brain. So the frontal lobe is more involved in your thinking, executive functions, and a lot to do with your efferent signals. So all your movement and stuff will be part of the frontal lobe, and the motor cortex is part of that frontal lobe. It's just the backmost part of the frontal lobe. So that's your movement. And then the sensations part of the parietal lobe. And the parietal lobe is your perception, making sense of the world, arithmetic and spelling. And they're either side of this line down the middle, and that line is called the central sulcus. Then your occipital lobe is involved in vision, and the temporal lobe is involved in memory, understanding, and language. So, coordination of movement. The cerebral, basically what happens in this very complicated diagram is that the cerebral cortex will send like a motor plan. So it says, maybe I'll move this muscle, then this muscle, and this muscle to try and do this action. So it sends that information to the basal ganglia and the cerebellum. And then the basal ganglia and the cerebellum say, okay, I'll probably modify a little bit to make it work better, a bit more coordinated. Then they send that message back, and the cerebral cortex is like, all right, sounds cool. And then it sends that to the spinal cord. So the basal ganglia and the cerebellum are kind of like they provide advice to the cerebral cortex. They modify signals to kind of make your movements better. The basal ganglia are mostly involved in planning, timing, and scaling of movements, and the cerebellum is more involved in like balance and coordination of movements. So the cerebellum, as I said, balance and posture, coordination, motor learning, which is like your learned actions, things you don't have to think about, like riding a bike, that kind of thing. And cognitive function is a little bit of cognitive function as well. So it takes information from the cerebral cortex via the pons. So the cerebral cortex can't contact the cerebellum without going through the pons, and then the cerebellum sends that information back via the thalamus. So the thalamus is like the relay center. Nothing gets to the cerebral cortex without the thalamus. The basal ganglia involved in planning, unconscious movements, timings of actions and scaling. They receive information directly from the cortex and then move it back via the thalamus. Let me just check if the recording's still working. Yeah, it should be still recording. Yeah, cool. Okay. So basal ganglia, all the different parts of the basal ganglia, it is really scary, but I'll try my best to break it down. Even at my level, they tell us we don't need to know the exact components, like very in depth, but I've got this really good diagram from a person in my year level. Hopefully it can help a little bit understanding the pathways. Basically, all you need to know is that the direct pathway promotes movement and the indirect pathway inhibits movement. So I'll kind of explain what happens in each. So the cortex, that's where the information starts. So it sends, or remember the cortex sends information to the basal ganglia. So it excites the striatum. Then the striatum inhibits the globo pallidus interna and substantia nigra reticulata. And that leads to disinhibition of the thalamus. So usually these two inhibit the thalamus, but if this is inhibited, then it can't inhibit the thalamus. So the thalamus goes ahead and excites the cortex. That's the direct pathway. And this is the indirect pathway. So the cortex excites the striatum inhibits the globo pallidus externa. Then since this is inhibited, the subthalamic nucleus is disinhibited. So it's free to go excite this, which inhibits this, which doesn't excite the cortex. So when you excite the cortex, you facilitate movement. When you don't excite the cortex, you inhibit movement. So indirect pathway leads to not exciting the cortex. And this is just another summary of that. You can take a screenshot if you want. I think this is a really good diagram. Basically, the way that we control which pathway we want to go down, basically they're both happening at the same time, but the way we control which one is stronger is that through the substantia nigra compacta, the D1 receptor will trigger the direct pathway. So it releases dopamine. The D1 receptor triggers the direct pathway. And the D2 receptor inhibits the indirect pathway. So overall, both of these receptors lead to increase in movement. And this is pretty much what I'm saying here. So the substantia nigra enhances movement by releasing dopamine, enhances the direct pathway via D1, and inhibits the indirect pathway via D2. So obviously, if you're inhibiting the indirect pathway, you're inhibiting inhibition. I hate those double negatives, but that's how it works. And I find that to understand all those double negatives, it really helps if you draw out a diagram, just like one of these, and just go through it one by one to try and figure it out. The cortex also inhibits movement, so it releases ACH to inhibit, or pretty much to do the opposite. But yes, cool. So the spinal cord. A good mnemonic to remember your sensory, afferent, dorsal, all that stuff is same Dave. Sensory is afferent, motor is efferent. So afferent, obviously stuff coming towards the brain, efferent going away from the brain. Dorsal is your posterior aspect, so that's afferent. Ventral is efferent. So anything in this area of the gray matter will be going towards the brain. This area will be going away from the brain. Cell bodies reside in the gray matter. This is where they synapse. So they travel long distances in the white matter, and then they come to the gray matter. They synapse onto a motor neuron here. So for example, if it's a corticospinal tract, it'll synapse in the ventral horn into a motor neuron. Then the lower motor neuron will travel through the ventral root and go to the muscles. Axons are in the white matter. Obviously, they travel long distances through there. All right, so I won't get you guys to actually draw out a full diagram here, but just have a think about what each of these, how you would label each of these components of the spinal cord. So like this is the dorsal aspect, this is the ventral aspect. Think about what is this thing, what is this thing, what are these thingies, and then I'll go through the answer. All right, so this is the dorsal horn. This is the lateral horn. The lateral horn is usually where your sympathetics synapse, or they might, or sympathetics travel through the lateral horn. Your ventral horn is where your motor neurons synapse. Dorsal horn is where your afferent neurons will synapse. Then you got your dorsal root. The dorsal root ganglion is where the cell bodies are. So if you think of your sensory neuron, your cell body is kind of along midway in the neuron. So it's not like motor neurons where the cell body is kind of at the end of the neuron. It's kind of midway in the sensory neurons. So you get this large ganglion of cell bodies here. This is the sympathetic chain, which runs parallel to the spinal cord. And it's kind of, because it doesn't have enough space in the spinal cord, it runs parallel. So your sympathetics will come out and they'll go into here, then they'll synapse, and then they might move out and go to some organ or something. And then once you get to this mixed spinal nerve, so these just have sensory, these just have motor. And then once they mix in here, and then your dorsal rami will go to dorsal organs, your ventral rami will go to ventral organs, so muscles and stuff. And these will have both motor and sensory, and these will have both motor and sensory. So these are the tracts in the spinal cord. So as you can see, they're not in the gray matter. These are the tracts when they're traveling long distances, they travel in the white matter. So your posterior columns here, that's for the dorsal column medial lemniscus pathway. Then you've got your anterolateral spinothalamic tract. So lateral spinothalamic tract and anterior spinothalamic tract. And then obviously they're on both sides, but they're just split in this diagram into right and left, are ascending and descending. Your lateral corticospinal tract is over here, and your anterior corticospinal tract is over here. So it's not too difficult to remember. DCML here, spinothalamic here, and then corticospinal is kind of split across. Motor neurons send signals from the brain to the muscles to cause movement. Quite simple. Upper motor neurons go from the brain to the, in the corticospinal tract specifically, they go from the brain, travel down to the spinal cord, then they might decussate, which means move to the other side, and then they synapse onto a lower motor neuron, which takes the signal to a muscle. All the muscle fibers innervated by one motor neuron is called a motor unit, and those act together. So all those muscle fibers will contract at the same time because they're all controlled by one motor neuron. Cool. So different types of motor neurons. There's alpha motor neurons, which control the extrafusal muscle fibers. Now the extrafusal muscle fibers cause movement. The intrafusal muscle fibers, they constitute a muscle spindle, and they keep the muscle spindle taut so that it's kind of tight, and therefore you can detect stretch. So it's more, it's an efferent muscle fiber, so it causes contraction, but it's not so much to contract the entire muscle. It's more to keep the muscle spindle taut so that so that we can sense how tight the muscle is. And the muscle spindle kind of is kind of a receptor in that sense. So the important ones are alpha motor neurons, which innervate extrafusal, and gamma, which innervate intrafusal. Force summation is how we get kind of more force from the same myofibrils or from the same muscle cells. So one twitch is when you get one signal and then it comes down again. So this is how much tension there is. If you kind of summate those waves, so it builds off each other and your tension gets higher. So the faster the frequency, the more tension you're going to get because you're building off the previous wave. Before it comes down again, you can fire another action potential and it increases the tension again. And if you get high enough frequency, the tetanus, so tetanus is like when you get very high frequency so the muscle stays contracted, it fuses. So the tension doesn't vary. It just starts becoming a straight line because your frequency is so fast, you can barely tell the difference between those waves. So the size principle, it took me a while to understand this, but basically, with these motor units with neurons with smaller cell bodies, they require a lower frequency of action potentials to excite it. The ones with bigger cell bodies, they require a higher frequency of action potentials to excite it. So that way we can control how much power or how much tension our muscle is exerting. So if we have a low frequency, these small motor units will be activated, but these will not. Then as we increase the frequency, we're activating X, we're activating Y, and then if you increase it even further, we're activating Z as well. So all of those will be activated. So for our fine motor units, motor actions, we only want to use X because we don't want to have too much force. But obviously if you're like lifting weights or something, you want to use all of these. So you'll start increasing the frequency. So the bigger cell body will be activated and you can use all of these muscles that it's attached to. Descending motor pathways, you need to know the difference between pyramidal and extrapyramidal, but the only tracts that you'd really need to know is the corticospinal tract. So the pyramidal descending motor pathways are the ones that control voluntary movement, and they move through the pyramids of the medulla. It's just a section of the medulla. Don't need to worry about what it actually is. It's just a little part of the medulla. The neurons move through it. So upper motor neurons directly synapse onto lower motor neurons in the spinal cord or the brainstem. Corticospinal tract controls like your body. Your cranial nerves, corticobulbar tract, control all the stuff in your head and neck. Extrapyramidal tracts control things like involuntary things like reflexes and posture. And we'll discuss a little bit about rigidity and spasticity, which can be used to differentiate between these two. So the corticospinal tract, I've mentioned it a little bit, starts in the primary motor cortex, travels down in the tract I was mentioning before, in the white matter tract. Right after the medulla, some of the fibers, like 90% of them, will decussate, which means they move to the other side. Some of them will still keep going down in the same path. And then once they're ready to exit, they'll move to the gray matter, synapse onto lower motor neurons, and then the lower motor neurons will carry that message out to the muscle. So there's only two motor neurons involved in this, upper motor neuron and lower motor neuron. Spasticity is when you get a lesion to your pyramidal tract, so your voluntary tracts. It's characterized by sudden, when you're testing tone, it's characterized by sudden increases and decreases in tone. So you might get like a clasp knife spasticity, which is when you have to push a little bit and then all of a sudden the arm just kind of jerks back. So it's the velocity dependent, whereas rigidity is with extrapyramidal tracts, and it's a constant increase in tone throughout the movement. So you might get cogwheel rigidity in Parkinson's, and that's still rigidity. It's a constant increase in tone, but it's kind of due to a tremor. So that's why you get the cogwheeling. In spasticity, in rigidity, you get equal increase in tone to both the agonist and the antagonist. With spasticity, it's mostly just one group of muscles, either the agonist or the antagonist are very hypertonic, where the other one's hypotonic. So upper motor neuron lesion signs. So this is how we can differentiate between damage to the upper motor neurons and with the lower motor neurons. Initially, straight after the damage to the upper motor neurons, you'll get spinal shock. So that leads to flaccidity, hypotonia, and areflexia, because your lower motor neurons are like, oh, what the hell? We just lost our upper motor neurons. And then they go flaccid. All your muscles will go flaccid. In the long term, the lower motor neurons kind of have a sense of what's happening. They lose innervation from the upper motor neurons. So it leads to loss of fine movements. So we can't control our muscles from our brain, or most of the muscles at least, or most of the motor neurons. It leads to weakness. So it's harder for us to control like the strength of our actions. It leads to spasticity, which I've mentioned, hyperreflexia and hypotonia. And I'll discuss why it causes like increased tone from damage to the upper motor neurons. And then it also leads to positive Babinski reflex and other primitive reflexes, because those primitive reflexes are actually inhibited by our upper motor neurons. So when the upper motor neurons get destroyed or they get damaged, those primitive reflexes come back. So why do upper motor neuron lesions cause hypotonia as well as weakness? Those seem like opposite things, but hypotonia is kind of your involuntary, it's kind of an involuntary reflex, right? So when you're moving your arm around, hypotonia will resist that movement, right? Weakness is your conscious control. I want to move my hand around. hypotonia is when you're moving the patient's hand around. Descending pathways either excite or inhibit lower motor neurons. Damage to upper motor neurons causes decreased excitatory signals to lower motor neurons, causing weakness. So that's when we want to control our limbs, we can't, because our upper motor neurons can't control the lower motor neurons. Upper motor neurons also usually inhibit the effector fiber in a reflex. So if we're looking at this reflex here, you might get the ankle jerk reflex. These sensory neurons come along, they come to the spinal cord, they synapse. So this would be a stretch reflex because there's no interneuron. The motor neuron, they'll synapse onto the motor neuron, and usually we get this inhibition from a descending tract. So usually the descending tract is like, okay, chill, it's not that big of a deal. You can decrease the efferent signal a little bit. If these descending tracts are not working, then we don't get inhibition of the reflex. So the reflex actually becomes a lot stronger than it should be. So you get an exaggerated reflex. And your tone is very closely related to your reflexes as well. So you get increased tone. So you might get muscle atrophy, hypertrophy, because your muscles are always working harder than they should be because there's no inhibition from the motor neurons. All right. Lower motor neuron signs. This is when your upper motor neurons are working. They're telling the lower motor neurons, okay, you need to move this muscle, but the lower motor neurons are damaged, so they can't. So that leads to permanent flaccid paralysis, or actually they might regenerate, but you get flaccid paralysis, you get weakness as well. So both cause weakness. You get muscle atrophy because your muscles aren't receiving any signals at all. And then you can also get areflexia. So it's very similar to the early signs of upper motor neuron lesions. Spinal reflexes, the 5 main components. You get a receptor, you get an afferent fiber, so that's the sensory neuron going to the spinal cord. You might get an interneuron for some of them, so that's with the Golgi tendon reflex and the withdrawal reflex. And you might also, then you'll get an efferent fiber, that's the motor neuron, and then the effector is the muscle. So these don't require input from the brain, but obviously the brain can inhibit these reflexes. The stretch reflex is basically when your muscle's getting stretched and you don't want it to stretch. So if you're collapsing or something, your quads might start stretching and you want to slow down its stretching because you don't want to collapse too quickly. So your muscle spindle will send, so we mentioned the muscle spindle and the intrafusal fibers. So those will be very tight. So when the muscle stretches, it'll send a signal to the sensory neuron back into the spinal cord. And usually it's monosynaptic, can also be polysynaptic apparently. But it is a bit of both. The motor neuron will come back and, the antagonist will be polysynaptic. The motor neuron will come back. It'll agonize the muscle that's being stretched and relax the muscle that's not being stretched. Okay, so that helps maintain balance. You don't have to think about it, because if you had to think about maintaining balance, that would require a lot of energy and it would take ages as well. Golgi tendon reflex is when your muscle's getting overworked. So it's the opposite. And so if your muscle's getting overworked, you don't want the muscle to tear, you let go. So you relax that muscle. So the Golgi tendon organs are in the tendons, obviously. And if they send a signal to your spinal cord and says, okay, we're about to get torn, it relaxes that muscle. And that's polysynaptic. The withdrawal reflex is when you get pain. So you get a sensory neuron going to the spinal cord. You get an interneuron, and then a motor neuron pulls or excites certain muscles to pull away from the painful stimuli. Autonomic nervous system. Well, this is all the stuff we can't consciously control. So sympathetic is fight and flight. Parasympathetic is slow down, rest and digest. The sympathetics run from T1 to L2. Those are where the spinal roots come from. And then your sympathetic chain runs a bit further than that. That's just where like your fiber might come out here, move up a bit, and then synapse, and then it'll move to whatever target it has. So the ganglion is where it synapses. And with your sympathetics, you'll notice that they don't move very far before they synapse, and then they move a long distance to the target organ. And that's kind of the opposite of your parasympathetics, which move a long way, then they synapse. They synapse very close to the target organ, and then the postsynaptic neuron will travel a short distance to the target organ. We also notice that the parasympathetics are craniosacral, so they'll come out of the medulla, brainstem, as well as the sacral portion of the spinal cord, whereas the sympathetics are from T1 to L2. So this is a table summarizing all the bodily functions, sympathetic and parasympathetic. It's usually quite self-explanatory. Or if you think about it, if you're resting, you want your, I don't know, maybe your stomach will increase contractions because you want to rest and digest. So you want to digest your food a bit more. So you increase contractions of your stomach. Yeah, so usually there's these things that are self-explanatory, but you can have this table as well. So this is a diagram showing you like the receptors involved in different pathways. So your first receptor is always going to be nicotinic. So with all of these receptors, the first receptor is nicotinic. Once it comes out of the CNS, it's nicotinic. So your somatic, your muscles, your skeletal muscles, that'll be nicotinic receptor from acetylcholine. And then your sympathetic cholinergic and your parasympathetics will be muscarinic. So sympathetic cholinergic is a bit of a weird one because it's still sympathetic nervous system, but it releases acetylcholine when it should be releasing norepinephrine or noradrenaline. And that's innervation of sweat glands and some blood vessels. And then obviously with your adrenergic sympathetics, you get nicotinic receptor, and then you get alpha or beta receptors receiving noradrenaline. So this is a quick summary of your autonomic receptors for the sympathetic nervous system. A1 leads to contraction. So you contract blood vessels, contract the sphincter in your digestive system, contract eye radial muscles, gluconeogenesis, that's increase in sugar. Obviously, you guys know that. A2 is negative feedback. So if you think of like a motor neuron like that, your A2 receptor might be here. So when you release the neurotransmitter, it'll come back, bind to this A2 receptor, and it says, okay, chill, don't need any more. So it's negative feedback. B1 is exciting. So with the 1, think of the heart, it'll excite the heart. B2 is relaxing. So it'll relax the lungs. So two lungs, one heart. So all the things that need relaxing will be relaxed by this. Exciting will be excited by B1. We do not use a B2 agonist in heart failure. So the B2 agonist will be used in asthma when a person doesn't have heart failure. And that's because if we give a B2 agonist to relax the lungs, then there is a chance of cross-reactivity, which increases activation of B1, which would be very bad for a person in heart failure, because in heart failure, your heart's working too hard. It'll lead to even more hypertrophy and exacerbation. B3, don't need to worry about too much. It's just related to lipolysis and gut muscle relaxation. So sweating is the weird one I was talking about. It's sympathetic innervation, but it's a muscarinic receptor. So it releases acetylcholine. So the parasympathetic receptors are M1, M2, M3. M1 is your salivary glands, gastric glands, and you also get a lot of signaling in the cerebral cortex with M1. M2 is decreased contractility of cardiac smooth muscle. And M3 is vasodilation and contraction of smooth muscle in the respiratory tract and gastrointestinal tract. So a cool way of remembering the parasympathetic effects is be my sludge, bradycardia, bronchoconstriction, meiosis, which is like contraction of the pupil, salivation, sweating, lacrimation, which is obviously tears, lethargy, urination, diarrhea, increased motility in the gastrointestinal tract, and emesis, which is vomiting. So the cholinergic junction is when you release acetylcholine. It's broken down by acetylcholinesterase in the synaptic cleft, and then it's also reuptaken by transporters in the presynaptic membrane. The adrenergic junction uses the neurotransmitter noradrenaline, and clearance is by breaking down by MAO and COMT. Those are those enzymes there. You don't really need to know what they stand for. Reuptake into the presynaptic neuron happens as well. And then you also get extra neuronal uptake. So that's into the blood maybe, or into another organ. So in terms of the male sexual response, it uses both the parasympathetic and the sympathetic nervous system. So the parasympathetic nervous system will release nitrous oxide, not acetylcholine, as usually happens. And it dilates the penile artery, increasing blood flow to the penis and causing an erection. And the penile artery is the only blood vessel with parasympathetic innervation. The rest are all sympathetics. The sympathetic nervous system controls the orgasm, so it leads to rapid contraction of the pelvic floor muscles. So that's all the muscles on the pelvic floor, obviously, and then that leads to contraction of the vas deferens and ejaculation. So emesis is vomiting. Anyone who's vomited knows what happens mostly. When you vomit, you get deep inhalation, closure of the glottis, and raising of the soft palate prepared, like food coming out of your stomach, suspension of breathing, and then your diaphragm and abdominal muscles are actually the things that contract, not the muscles of your stomach. So those muscles contract, they squish the stomach, all your stuff comes out of the esophagus to get ejection of gastric contents, and that might repeat. So there's four main things that can trigger the vomiting center, which is in the medulla oblongata. So the vomiting center controls vomiting, controls all these things involved in vomiting. So you might get anticipatory emesis. So that's coming from the cerebral cortex. You might look at something really disgusting and you want to vomit, and that will trigger the vomiting center. Vestibular center controls balance. So when you're very dizzy, you might get vertigo. That'll trigger vomiting, so motion sickness, that kind of thing. Your peripheral receptors, so receptors from the rest of your body. So if you've got irritation in your stomach, irritation in your intestines, you might want to vomit as well. And then the chemoreceptor trigger zone just detects different chemicals in your blood and may trigger vomiting. So stuff like opiates and anesthetics. And vomiting is also preceded by profuse salivation. So that would be parasympathetic stuff. Cool. Okay. So I'll get you guys to maybe type in the chat what you reckon for this answer, A, B, C, or D. Where does the corticospinal tract decussate? For some reason, I can't see the chat. Oh, there it is. Good. Just off the medulla in the spinomedullary junction. Very good. The neurotransmitter between the presynaptic and postsynaptic neuron in the parasympathetic nervous system is what? Cool. Acetylcholine. So noradrenaline is used in the sympathetic nervous system onto the organ. And yes, James, technically 10% do decussate at D in the spinal cord, but most of them decussate just after the medulla. You're cooking noodles and some boiling water gets onto your hand. Which of the following would occur? First, sorry. Yeah, good, C. Stimuli is transduced into an action potential. Okay, sensation. Sensation is the conscious or conscious detection of stimuli, so that's happening over here. Perception is how you, like your interpretation of the pain. Is it that bad? Is it, what kind of pain is it? That's your perception of it. So there's four main sensory fibers involved in sensation and perception, and these last two are involved in pain. So it's kind of easy to remember. Your myelination, so you go A alpha, A beta, A delta, and then C. Your myelination decreases as you go down to none at C. Your diameter decreases. Obviously, these ones will be faster. These ones will be slower. A alpha is involved in proprioception, so that's awareness of where your limbs are in space. Mechanoreceptors are controlled by A beta, and A delta controls pain, and C controls pain as well. So these are your types of receptors. You guys should know most of these receptors already. Your nerve firing can be phasic or tonic. Phasic is when it's fast adapting, so it adapts very quickly. So there's a short burst of action potentials, and then it quickly kind of slows down. And then tonic is it's very slow, gradually increases in frequency, and then it maintains that. So that way you can provide information about the duration of some kind of stimulus. 2-point discrimination is the ability to discern the difference between two nearby objects and discern whether they are truly two distinct points. And it's controlled by how dense the density of mechanoreceptors in that area. And obviously, the more dense it is, the better resolution you have. So these are the mechanoreceptors that they want you to know about. Pretty much, so I remember it, MRMP, Mr. Map, your slow adapting MR, fast adapting MP. These ones are your non-hairy, these ones are your hairy. And then these two are touch, the top two are touch, and then the bottom two are stretch and vibration. And this is just a summary of those. I realize I'm running a little bit short of time, so I'll speed up. You can have a look at this on the slides. Proprioception is awareness of the orientation of your limbs in space. So they use muscle spindles, Golgi tendon organs, and joint receptors to be aware of your limbs. The dorsal column medial lemniscus tract is another tract that we need to know. So it's the sensation of fine touch, proprioception, and vibration. Those are the main things that the dorsal column medial lemniscus tract can senses. So basically what happens is your first order sensory neuron comes in through the dorsal root, travels up into the nuclei of the medulla, and then synapses, second order, will decussate into the medial lemniscus, and then move up, synapse in the thalamus, and then the third order will go to the primary somatosensory cortex. And this is where we perceive the information in this third order. The spinothalamic tract senses temperature and pain. Those are the main two. And it's similar, except the spinothalamic tract is the only tract that we need to know about that decussates almost right after it enters the spinal cord. Whereas the other two kind of decussate around the brainstem. The anterior tract controls crude touch and pressure, not as important as pain and temperature controlled by the lateral tract. And this is just showing, obviously now we know, spinothalamic, DCML, corticospinal. Okay, you guys have a go at this question on the pre-slides, on the post slides. Yeah, and message me if you have any questions with that. Nociception is the perception of pain. Chronic pain is when the pain doesn't serve like a physiological or evolutionary purpose. There's 4 phases. Transduction phase is when we convert the noxious stimuli into, or it's activated by activating nociceptors. Transmission phase is when we kind of take that information and bring it to the brain. The modulation phase is like we're editing the pain signal. So we're deciding whether we want it to be very impactful or we don't. It's like the pain's not that bad. And that's controlled by descending neural systems. Perception phase is our awareness of that pain. Transmission of pain, as I mentioned, happens through A delta and C fibers. C is more like the long-term aching, burning, dull pain. A delta is the sharp pricking pain in the short term. The gate theory of pain, this is when, like, say you scratched your arm on like a twig or something. If you rub that part of your arm, you get lots of touch sensation going up to your brain. So that touch pathway, so the mechanoreceptors, will inhibit the pain pathway. So the signal actually gets a bit weaker. So the pain signal gets weaker because we want to focus on the touch rather than the pain. So pain modulation, it also happens through these things called enkephalins, which is from a descending pathway. So interneurons will release enkephalins, which inhibit second order neurons. And I'll show you what that looks like on the next diagram. Opioids will mimic enkephalins, so they'll decrease the pain that you perceive. So these descending pathways activate this interneuron, which releases enkephalins, and it inhibits the second order neuron. So pain doesn't, it should be this one. Pain doesn't go up to the brain, or decreased pain will go up to the brain. Central sensitization is when we get so much pain, like chronically, that we become more sensitive to it. So there's a lower threshold for pain, and it's characterized by hyperalgesia, so increased sensitivity, and allodynia, it's just pain from non-painful stimuli. So which of these is found in hairy or thin skin? All right, so it should be A and C. Just think of pet ruffles for your hairy. So think of a hairy dog, pet ruffles, Ruffini's and Pacinian. Where does the tract carrying vibration and proprioception decussate? Good, it's the medulla. The spinothalamic tract is the only one we need to know about that decussates in the spinal level. Nothing decussates in the thalamus, and nothing decussates in the primary somatosensory. What are the correct order of the pain pathway? Yep, so this one should be B. What color is the first order nociceptive neuron? I think that's kind of brown or red, however you interpret that color. Yeah, cool. Where is its cell body? It's in the dorsal root ganglion. Its synapses in the dorsal horn. Which order neuron has its cell body in the dorsal horn? Should be second. What effect will activation of the blue receptor have? All right, should be D and E. So what activation of the blue receptor will lead to that gate theory of pain. So the excitation of this inhibitory neuron, which decreases the amount of pain going back to the brain. And that would be a Pacinian corpuscle, which looks like an onion. What effect will excitation of the green neuron have? So that was actually D. So you're inhibiting the pain reception or the pain pathway by inhibiting a second order nociceptive neuron. And the funky gray thing, that's just a motor neuron involved in the spinal reflex. Yeah. Where does the spinothalamic tract decussate? Yeah, C, awesome. Good, you guys are listening. All right, cool. Types of muscles. You guys have done this to death probably. So you got cardiac muscle, skeletal muscle, smooth muscle. Skeletal muscles are the only ones that are voluntary. Skeletal muscle, striated. You can see all these sarcomeres lined up over here. They're multinucleated, so they have lots of nuclei because all these cells have come together to form one cell. And then you get nuclei in the periphery. So that's differentiating from the cardiac and smooth muscle where the nuclei are more central. And we'll have a look at that here. So the central nuclei here, and there's also branching, which allows the movement of the action potential like in a three-dimensional space. And obviously the cardiac muscle would be involuntary. Smooth muscle, you get central nuclei as well. It's non-striated. You can't see any sarcomeres here, and it's involuntary. Okay, so A will be your skeletal muscle. We can see the nuclei in the periphery of the fibers. We can't see striations, and that's because the muscle's coming at us out of the page. This will be cardiac muscle. We can see the central nuclei, and as we can see a little bit of branching as well. And there's this obviously smooth muscle because there's no separation of any of the fibers. So we can probably skip over that. The skeletal muscles are arranged such that the sarcomere is made up of thick and thin filaments. The myofibril is one long fiber of lots of sarcomeres joined together. A muscle fiber is a lot of myofibrils joined together in one cell. So the muscle fiber is one cell. You wrap a lot of, and that's wrapped in endomysium. You bring a lot of muscle fibers together, you wrap them all together in perimysium, and that's a fascicle. Then you bring a bunch of fascicles together, you wrap them in epimysium, and that's a muscle. So in the muscle cell, we've got the sarcolemma, which is the plasma membrane. The transverse tubules are invaginations of the plasma membrane into the cell, and that just allows propagation of the action potential into the entire cell very quickly. The myofibrils are all these sarcomeres stacked onto each other. The sarcoplasmic reticulum is like the endoplasmic reticulum. It stores all the calcium. So, oh, sorry, that's the T tubule, my bad. This is the sarcoplasmic reticulum, stores all the calcium for quick release when it's needed. And a triad is the sarcoplasmic reticulum, transverse tubule, then sarcoplasmic reticulum, all lined up like that, as opposed to a dyad, which in cardiac cells will just be sarcoplasmic reticulum, then T tubule, and then you might have sarcoplasmic reticulum, T tubule, but then they're not all lined up. So that's a dyad. The sarcomere, the A band is all the thick filament. The I band is just the thin filament. The H zone is where the thin filament doesn't overlap with the thick filament to get that clear space in the middle. The M line, think of M, midline. Z looks like a Z. That's a Z line. The filaments, okay, so actin has all these binding sites for myosin, and tropomyosin kind of covers those binding sites. Now, when you release calcium into the cell, the calcium will bind to the troponin, and the troponin will pull the tropomyosin away from the actin and reveal those binding sites. So with the binding sites revealed, with the myosin can bind to the actin and start contraction. So cross-bridge cycling, this is a difficult concept, so I'll try and explain it to the best of my ability. Basically, in order to get it started, we need ATP. So that's why when people die, their muscles get very rigid, because ATP allows the myosin to detach from the actin. So ATP attaches to the myosin head detaches from the actin, and that kind of relaxes the muscle. And then ATP is hydrolyzed to ADP and PI. Then we release calcium when we want to contract the muscle. So the myosin will bind to the troponin, myosin will bind to the actin because the calcium allows those binding sites to be revealed. Then the ADP and PI will be released, which causes a power stroke. So the myosin strokes and pulls the actin closer to the center. And then you might get ATP binding again, and that'll bring the myosin back. So it clocks the myosin back. The ATP will be hydrolyzed to ADP and PI, and it starts the whole cycle again. So physiologically, the physiology of contraction, your action potential comes down through the neuron. It'll go through the transverse tubule, the T tubule. Then the T tubule will activate some receptors, which lead to the sarcoplasmic reticulum releasing intracellular calcium. And that intracellular calcium will reveal the binding sites on the actin, and that'll lead to contraction. Then once everything's kind of cleared up, your intracellular calcium goes back into the sarcoplasmic reticulum for storage, and all the contraction ends, everything relaxes. So there's two main types of contractions. There's isometric, when it's the same force and the length of the muscle does not change. But isotonic is when there is a force, but the length of the muscle changes. So concentric is when there's a force, but the muscle shortens. So that's like your bicep curls, you're curling the muscle, you bring it up, that's concentric. And then when you're letting it down, that's eccentric. So there's a force, but it's kind of slowing it down rather than contracting the muscle. So there's three ways of controlling contraction strength. So there's the length of the fibers, there's the frequency of action potentials, and there's the number of motor units recruited. So in terms of the length of the muscle fiber, if there's a lot of overlap here, there's not much space that the sarcomere can actually contract. So it can't develop much force. If you need this, like a decent H zone for contraction to occur, because obviously if you got like two ends of the sarcomere like that, you can't really push in to contract the sarcomere. But if you got space like that, there's space for it to contract. But if you got too much space, there's not much overlap between these heads and the actin. So not as much cross-bridging happening. cross bridge cycling happening. So there's not as much force being generated if the muscle's too stretched. Force summation, we've already mentioned, so I won't go through that. Motor units, we've kind of already mentioned. There's an all or nothing principle, which I've mentioned. So if one motor neuron is activated, all of its muscle fibers will be activated. All right, what is F? Cool, F is the sarcomere, very good. What is G? G is C, very good. What is J? J is the A band, very good. What is I? I is the H zone, awesome. Okay, so the way I kind of remember those is that I is a very thin letter, so it'll be the thin part of the sarcomere, so thin filaments. A is a bit thicker, so if we're just looking at A band, I band, I mean the A is a bit thicker, so it'll be the thick filaments. I know it's a bit of a crappy way of remembering it, but it works. The H zone is the only zone there is, so that's going to be the one in the middle. M line, obviously midline, and Z line, it looks like a Z. Cool. And the last question, what's the best medical TV show ever made? You guys better answer this correctly. Yes, correct. It is Scrubs. I was scared people were going to say Grey's, but Grey's isn't even second. House is second. Okay, cool. Does anyone have any questions? Oh, thank you to Kayla Robus, who did this lecture last year. I've taken a lot of content from the slides from that. Please email me if you have any questions. Are there any areas that people want me to redo because I might have gone too quickly because didn't have much time for questions? Or any areas anyone's unsure about or any questions anyone has?
Oh yeah, could I just ask? It takes a long time to write down, so can I just speak?
Yeah, of course.
Just in terms of like the role of the hypothalamus and the thalamus, in terms of that sort of part, how much should we know about that?
Okay, so the thalamus, as far as I know, and I'm like at a second year level, thalamus is just like the relay center, so it controls. Well, it doesn't really control much. Like all the signals will go through the thalamus from the brainstem to get to the cortex. So it's kind of like, so if you got the cortex here, you got your thalamus, and then you got your brainstem and cerebellum. It has to go through the thalamus to get to those things. It's the relay center. Hypothalamus is involved in your homeostasis. Okay, and hopefully that answers your question. And hypothalamus kind of plays a role in the pituitary gland and all that stuff as well. Okay, someone's asked, could you explain a little bit more about the indirect and direct pathway? Okay, I'll try. So I stress a lot about the indirect and direct pathway. Don't need to stress about it. The main thing is obviously, as I said, direct pathway increases movement, indirect decreases movement. So, and like once you understand disinhibition, it's a lot easier to understand. So disinhibition is when you inhibit the inhibition. So if think of it like a bruise bus, you're inhibiting people moving through, and then if you say, don't worry, no one's getting drunk, the bruise bus will kind of be chill. So you're allowing people to go through. That was a really shit analogy. So pretty much direct pathway starts in the cortex, excites the striatum. Then the striatum will inhibit these two here. Don't need to remember the names, GPI and SNR. And then because it's inhibiting these, they usually inhibit the thalamus, but since they're being inhibited, the thalamus is free to excite the cortex. The indirect pathway inhibits movement. So you get excitation of the striatum, which inhibits the global pallidus externa. And then since the GPE can't inhibit the subthalamic nucleus, the subthalamic nucleus excites these two, which inhibits this, which can't excite the cortex. So it leads to inhibition of movement. Oh, and a good way of remembering which ones are inhibition, which ones are excitatory, is that everything related to the cortex is excitatory, so only the stuff related to the cortex is excitatory, as well as the STN. Everything else is inhibitory. Okay, so cortex stuff is excitatory plus STN. Everything else is inhibitory. And we can see that here. So cortex stuff, excitatory, and plus STN. The rest is inhibitory. And then your substantia nigra will excite the direct pathway and inhibit the indirect pathway. All right, any more questions? And Zara's got a good mnemonic there, dark band, DA, and so that would be the A band, and light band, LI, and that would be the light band, obviously. Cool, very good. Any more questions?
Thanks so much, Ethan.
No worries. We'll pass it on to the next person.
Yeah, hey everyone. Sorry for the background noise. There's construction going on where I'm at. But I think we have Jan from MIPS, our sponsor here. So Jan, you can share your screen whenever you want.
Hi guys, how are we?
Doing well, thanks Jan. How are you?
Yeah, good, thank you. Thanks for having us along again today. I'm just checking that you can see my screen.
Not yet.
Okay, just bear with me 1 second, apologies. Does that one work?
Yep.
Perfect. Hi, everybody. Good morning. I'm Jan from MIPS, and thanks for inviting us along again today. I popped in and spoke to the group very briefly yesterday morning. So I have the same role as your regular MIPS liaison, Tamar, but Tamar got deployed into our finance group while COVID hit. So my role is based, I look after Perth over here in WA, and I also look after South Australia. So for the first time since February, I get back to go to South Australia next week and catch up with my students and my junior doctors over there. So I just thought, while the guys asked us to come along and say hello today, I ran a little competition yesterday, which we're going to keep open for you guys today. And I think it's going to be shared maybe on your Facebook post or between yourselves till close of business tomorrow. And then winners will be announced on Friday. So we've just put a little fun pack together. So what you'll need to do is scan the QR code or use the link there on the slide. That would just also let me check your details, because Tamar's not been on site very much this year. We just need to make sure that everybody's up to date, ready for next year. So that'll just be me looking after it for now. And hopefully by the time we start next year, Tamar will be back with you all. So just some things I was going to touch on as I did with the group yesterday. And I know I try and remind my students whenever I see them. Please don't forget our clinico-legal team is open to you guys as students. If you've got any worries about next year, or you're going on placement and you need any support while you're away, that's what we're here for. So our clinical legal team are very used to looking after students. We want all our students to feel safe and supported and not have any worries or stresses on top of our day-to-day living. So if next year you do start on a placement, or you're going off volunteering for your medicals, we're here all the time. So please don't worry if you've got any concerns. Please ring them and let them support you through your journey. We've also launched a lot of extra education onto our website just recently. During COVID, we've engaged with a lot of our doctors via this media, and they've found it very helpful. So there are some really interesting case studies. There's some webinars that you might be interested in preparing for the rest of your uni years ahead and entering into the hospitals on placements. And because we haven't been on site, I'm happy to take any questions from you guys. If anybody's got anything they want to share through chat or ask out loud is always good. I'm happy to answer those if I can today. And if not, the guys have my email address and I'm more than happy for that to be shared. I know student life has been really crazy, especially the lead up to exams. So if anything else today, look after yourselves and be kind to each other. And most of all, be kind to yourself. It's a stressful time, we know. So if we can do anything to help with that, please, we're here to do that for you. So scan the QR code or use your little link there, and hopefully we'll have some winners from you guys on Friday. And I will personally give you all a quick call if you're a winner, and then I will post the packs out from Perth on their way to you early next week. So has anybody got any questions while I'm here at all? No?
Thank you so much, Jen. I'll be sure to share your email in the QR code on Facebook tomorrow.
Yeah, lovely. And then on Friday morning when I wake up over here, which will be way past breakfast time over there, I'll contact whoever's been the winner and we'll organise getting those gifts over to them. And like I say, if there's anybody that needs anything else, I'm happy for you to share my email and my phone number. Everything is good. We're here to support while we can't be on site with you.
Well, thank you so much. I'll be sure to post all that info on the Facebook page.
Oh, hang on. I've just got a question. So if for any reason you're not sure, because it was a crazy year when we all started and you joined MIPS and hopefully got your water bottles, if you click the QR code today, your details will come through to me, and then I will check in our system and I will send you a screenshot of your MIPS profile and also a link should you wish down the track to update it. So if you move house or change your email address, it's always good to keep your profiles with whichever indemnity provider is looking, well, hopefully we're all looking after you, hopefully with all Avant and MEGA and MDA. So if you send that to me, I can have a little look for you and send you a link to update in the future. If not, you can always check back through your emails way back in January when you will have joined us. You can have a look through that or just visit the MIPS webpage and you can have a look at those informations if you can find your MIPS membership number in your old emails. But I'm more than happy for you to send them to me and I'll check. So scan the code, I'll pick up your details, and I promise we don't bombard you with emails. It will just be me sending you a link and some helpful information as well. So yeah, all good. All right, so thanks again for having us along. Good luck with exams. Stay safe, everybody, and we'll see you in 2021.
Thank you.
Thank you. Take care, everybody. Bye-bye.
All right, everybody. We'll take a small break and be back in about 15 minutes or so. So see you guys then. Hey guys, sorry, there's some issues with the Zoom, so give me 5 minutes to sort it out. Sorry. Hey guys, sorry, there's some issues with the Zoom, so I'm just gonna restart the Zoom, and then I'll try and restart it out. So if it drops, don't worry, it'll just be back up in a minute or two.
Hold on. Yunji, are you there?
Yeah, I'm here.
Okay. If possible, can you end the meeting for all? I think I made you host,
Cool,
and then
cool.
I
Yeah,
can
I'll
restart
do it now.
meeting. All right. Thank you.
All right. See ya.